Okay, so the second topic we have is genomic variants, and they come at many different levels, which is important to recognize, but not all of these things are so easily workable with bioinformatics methods. So if we take the largest and grossest kinds of variants, so we have the chromosomal anomalies, which can be detected by cytogenetic methods or staining of chromosomes spread from cultured cells. And these things were known about 100 years ago. So it was, or at least, let's say, 70 years ago, in 1950s, they did cytogenetics, and they knew that some diseases had an association with chromosomal abnormalities. For instance, the Down syndrome with trisomic chromosome 22 having 3 copies of one chromosome instead of the normal. There are some monosomies, and then we have insertions and deletions or translocations or changing bits of chromosome material, which can be detected by the staining. And because we get a specific band pattern in each chromosome to helping to recognize them, in addition to sorting them by their sizes. So if the band pattern is abnormal, we might see that some of the bands are as if in a reverse order, and that is a result of an inversion, so that a piece of the chromosome has been moved from up to down direction to down to up. And most of the time this is quite okay. The user does not suffer from it. The same genes are there unless the breakpoint for the inversion happens to break a specific gene. But there might be problems for fertility, but let's not go in that direction. Then we have something which are called structural variants, which are submicroscopic but fairly large, like 1,000 to 1 million base pairs. And these are things we definitely see at the sequence level, and the quantity of such variant has become obvious when we have more and more sequence data. One of the very early personal genome sequencing efforts was done for Craig Venter, the leader and founder of one of the genome industry companies, who was competing with the publicly funded project to arrive in a complete human genome. And actually it was not fair, because he was using the mapping data and other results from the public project to go faster with his shotgun sequencing, but he was not sharing his data with the public project. And the end result was that both projects ended more or less at the same time, and they made a joint publication with all. But anyway, to go back to the personal genome of Craig Venter, it was sequenced at such a high coverage that they were able to assemble separately the paternal and maternal chromosomes, so that they got the complete haplotypes, that is the sequence of the variants coming from different parents. And even if both of his parents are of Anglo-Saxon origin, moved to America sometimes like 200, 300 years ago, the other one was from Scotland and the other one from Ireland, if I remember correctly. But still, even between these two people from a fairly homogeneous region, there were multiple megabase scale inversions, large duplications, large deletions, and many more single nucleotide differences than was thought to exist between all of the human population. Okay, the structural variants might include the same as in the larger scale things, insertions, deletions, and inversions. And then copy number variations, well, that's more like having multiple insertions of the same region, or having multiple duplications of the same region, maybe the same gene. Then we'll discuss mobile elements in a minute, and adding those mobile element copies is also structural variation. It could be like a kilobase or a few kilobases. And it's notable that there are many kind of hotspots that seem to be more easily the starting and ending points of inversions, or positions where you might have inserted new sequences. And then the SNPs, or single nucleotide polymorphisms, that's what people usually think when we think of variants. So changing one letter in the DNA into something else, and that's your SNP. But then sometimes the SNPs can be thought to be simple nucleotide polymorphisms, so that very short indels, adding 1, 2, or 3 nucleotides in the DNA might be counted as SNPs as well. Okay, most of the time the copy number variations are just the difference between 1 or 2 copies, and in a way, a technical term for getting 2 copies of a gene from one parent would be partial uniparental disomy, so that there are 2 copies, and if there is no compensating deletion in the other parent, there might be a trisomy for a specific gene, which might be just okay. But trisomies of a whole chromosome are not good, because when you have an excessive gene dosage of hundreds of different genes, or maybe even 1,000 genes, so it's going to complicate too many things to be compatible with normal development, and not having any serious diseases. And sometimes, especially in cancers, we may see copy number variants that carry larger numbers, like even 10 or more copies of a single gene. And then we talk about gene amplification, and if the amplified gene is something that is related to cell growth or growth regulation, we might have a mechanism for abnormal growth behavior of the cancer cells. So just as an image, if we have 3 copies of some region in the reference genome, we might have gene gain or gene loss to have 4 or 2 copies. And inversions in a graphical form, so that you remember just changing the original direction for a fragment of DNA, which could be thousands or millions of bases. And translocation would mean moving a piece of DNA into a new position. Now, cancers are really horrible if you look at the chromosomes, especially in advanced cancer, when we have suppression of DNA repair mechanisms, which is one way of getting cancers. If the DNA repair is suppressed, so then abnormal genotypes can start living a normal reproductive life, and the cells proliferate. Whereas normally when we have DNA damage, the repair mechanisms either fix the problem, or if it's not fixable, if it's too extensive, they lead the cell to apoptosis or programmed cell death, so that the consequences of the abnormal genome would not harm the organism, would not harm the human. But anyway, once the mechanisms are suppressed, so we can get all kinds of translocations. So you can see that pieces of chromosome 2, some of them are in the chromosomes classified as 2, but then you have some red here all over the place. Well, curiously, chromosome 20 seems to be the only chromosome pair which is almost unaffected in this example. But we get all kinds of deletions, like there are no 2 copies of chromosome 5 that would have the correct length. Likewise, 1 copy of chromosome 12 is just 1/2 there. And then we have 1 piece of chromosome 12 here in chromosome 22. If you are interested in how people get these images, so you need to Google for fish, not the animal, but fluorescent in situ hybridization. That's the technology. Okay, let's talk about retroposons. You may have heard in genetics of transposons, which are mobile genetic elements, which can either move themselves into a new position, or they can make a copy of themselves and insert a copy in a new position. Some of these transposons can carry a load of other DNA, so that it can actually lead into moving pieces from one place to another, or creating duplications, even large-scale duplications like megabases. Now, these transposable elements have an RNA intermediate, and for that reason they are called retro element, because they are kind of reverse copied from RNA into DNA to make changes in the genome. There are some retroposons that are autonomous, so that they themselves code for proteins which are needed to create the transposition. And then there are other retroposons which have lost the capability of managing on their own, but because these mechanisms are active in our genomes all the time, so they can hitchhike on the machinery that is already there. And because of these retroposons, like doing their work behind the scenes, they have accumulated to a fairly high percentage. We'll have a graph of how much of them there are. And this I'll skip, so just to give you the names of some types. We have L1 elements, which are several kilobases, and then which are long interspaced nuclear elements. And then we have shorter versions that are less than 1 kb and are normally not autonomous. And one group of these is called the so-called ALU sequences, which come up as millions of copies in our genome. And an interesting statement I read in an article describing these retroposon elements is that the ALU sequences accumulate in GC-rich regions and L1 sequences accumulate in GC-poor regions. But this is a chicken and egg question. Do we have the sequences accumulating near other ALU regions because they already have some retro element recognition sequences there? Or is there an original start of GC-rich and GC-poor? But the fact is that the ALU sequences themselves are GC-rich, and the L1 elements are AT-rich or GC-poor. Okay, and these are the names of the elements in human, and in other species we have other kinds of retro elements. And it's interesting that not only these retro elements in the genome, we also have retroviral remnants. Retroviruses have a life cycle which includes copying their genome into the host genome and then having the host genome to transcribe and translate them so that new viruses are born. And these retroviral inserts have been accumulating in the human genome for a long time, and there are really many copies. And some of them are no longer mobile, so that they are just frozen there and changed beyond their moving functionality. And then we have some active elements that are still making new copies. And a nice fact is that 1 out of 600 new mutations are new L1 or LU insertions. And there are documented cases where a disease has its origin of having a new L1 or LU insertion within an important gene, so that we have a broken copy in the child, if this insertion happened in the germ cells of one of the parents. So you might have that. Okay, here's the graph I promised. So the surprising fact is that 45% of the human genome is comprised of these transposon or retro elements. We have the long and short elements, which are huge chunks, and then about 8% of these human endogenous retroviruses. We have other kinds of duplicates like short satellite repeats, and then we have segmental duplications. And the amount of protein coding genes is just 1.5% if you count the exons, but if you include the introns and the regions coding for untranslated regions, so the scope of the protein coding genes goes up to 30%. And there's a lot of stuff for which we don't have a specific explanation, not a function, nor the origin from the retroviral or retrovirus-like copying elements. And there's an old saying that garbage you throw out, junk you keep. So there would be an advantage for human cells. They would be replicating faster if we didn't have all of these retroelement copies. And it would be advantageous to throw them out if they didn't have any functionality, any purpose. But because they have stayed there, so maybe there is something important there. And there have been single reports of some specific L1 element being actually active in the brain, being transcribed at a specific point of brain development, and possibly leading to regulatory RNAs that are helping in the brain development. Let's see, we have something in chat. A reference where this figure is from, I think this is from a genetics textbook. It's really bad of me to take a picture and not quote the source there. I can't really remember which one. I'll try to look it up for later on. But if you look up human retro elements or human endogenous retroviruses, so there's been a lot of study of these HERV things, so that all the thousands of copies we have in the genome have been classified into families, so we can tell what type of of a retrovirus they have been originally and how they have evolved in the genome, which copies are old, which copies are more recent, and so on. And then there was a question of the cancer chromosome, cancer genome, chromosome 8. Oh yes, you are right. It's another chromosome pair that seems to be pretty intact, so that we don't see other colors there. Okay, this is the point where we could be looking at the ensemble variation slide set. Now, some of the material is duplicated, but I thought that the introduction, the biological introduction there is a little bit too brief, so I wanted to go to the background of variants a little bit deeper. But repeating is never a bad thing. So they say that it's really the mother of learning. That's like an old Latin phrase, repetitia mater studiorum est. Or should we say that it's the mother of studying? Well, in the old times, people focused much more on memorizing things, and there you obviously need the repetition. But the same applies for understanding any complicated concepts. When you approach the same concept from two different directions with two different forms of presentation, it helps you to learn them in a more stable manner, to remember them for a longer time. Okay, now we talked about the kind of classification by size, but here we'll also talk about classification of variants based on the effect, and we look at where ensemble variation data comes from, and then check the kind of things we have in ensemble for browsing the variation data. Okay, so here we have some examples of these small scale variants, minor insertions or deletions, and single nucleotide changes. And we have the large scale variants here. Here we have an example of a reciprocal translocation, so that pieces of chromosome 20 and chromosome 4 are interchanging. This is something that could happen due to non-homologous recombination during the meiosis of cells, so that we have pieces of non-homologous chromosomes binding to each other and leading into a crossing over event and the change of material from different chromosomes, instead of just changing material between maternal and paternal copies of the same chromosome. Okay, now let's look at variation consequences. Now, depending on where in the gene region we have the variant, now we are talking about single nucleotide variants mainly. So the consequences might be different. So this would be an example of a gene shown in the ensemble notation of having the protein coding exons here as bars, yellow bars in this case, and then the white pieces are the non-coding parts of the of the transcript. So we have the 3' untranslated region, 5' untranslated region here. And then we have the regulatory region or promoter somewhere upstream or on the 5' side of the coding region or the transcribed region that would help in getting the transcription started by having the RNA polymerase binding here and starting the transcription. And so if we have a change in the DNA sequence in the coding region, then the consequence might be a synonymous substitution, meaning that we get the same amino acid in the translation, or it could be non-synonymous or resulting in a change of the amino acid. All the terms for these things are silent mutations for these synonymous substitutions, or point mutations for the variants that actually change the amino acid sequence. And obviously the non-synonymous substitutions are more likely to have a biological consequence because the protein changes. But what about synonymous substitutions? Any of people with biology background, could you name some possible consequence for a synonymous substitution that is changing somehow the protein product or the biological behavior? So effects of synonymous substitutions. Well, there's a marginal chance that we might have a change in the speed of translating the transcript. Not all codons were created equal, so that even if we have 4 synonymous codons, we might have some, and especially if we have 6 codons for some amino acids, we might have some rare codons there that don't have the corresponding transfer RNAs for putting on the amino acids in the same amount. So changing into a rare codon could lead into slower translation. And likewise, for many household genes that have high expression, we can see that the message has been optimized so that it's using the most frequent codons all the time, so that the translation is not a bottleneck there. And then if we think about microRNAs, which we'll talk about later, microRNAs can bind to messenger RNAs by having a complementary sequence. And now if we have a synonymous change here, it might change the fit between a microRNA and the target here, so that we might either have a stronger regulation by a microRNA or a weaker regulation by a microRNA because of the sequence change. So we have mechanisms that could lead into a lower expression of the protein, even if the protein is the same in sequence. But this is like minor details. Don't worry if you don't think about these things every time you see a synonymous substitution. Most of the time you can just keep it, okay, synonymous. It's not going to be a reason for a disease. And these are extremely rare cases where where we might see something, the effect might be just a marginal individual difference, not a real disease-causing thing that would just stop the translation of a protein. But then changes in the splice site might be a big thing. We have specific sequences which regulate the splicing, or let's say guide the splicing. We usually have dinucleotides AT and AG, which are in both ends of the intron. And if we change them, the splicing mechanism might not see them, and the splicing might happen in the next AG somewhere else, or we might miss splicing at all so that we have a retained intron in the sequence. And obviously, that might be really harmful. Regulatory variants are interesting, but still not very heavily documented. We don't have a lot of them. When the genome was sequenced, it was thought that, oh nice, now we can really see the regulatory regions, how they work when we observe the variants that have an effect on the gene expression. And then we'll learn a lot about the variation, and we have a way of finding new mechanisms for diseases, but they haven't been so really prominent, and definitely not common disease mechanisms. So the most serious problems to protein function come from the non-synonymous substitutions, or sometimes if we have a if we have an amino acid coding triplet here changing into a stop codon, which means that the translation will be terminated. We'll get a truncated version of the protein, which is very often non-functional or with decreased functionality or maybe decreased stability, so that the protein is degraded, it doesn't fold properly or whatever. So the coding variants are, and the non-synonymous variants are still the most heavily investigated things. And regarding intronic variants or variants which are downstream or upstream of the gene, they are the variants that are most likely to be easily tolerated, so that we might have some difference to the splicing efficiency for some intron sequences which are not the real splice site sequences, but a little bit further in. But again, I'm going to find details. Just remember that not all variants have equal consequences, so that some of them lead to changes in the protein and some of them don't. And now the consequence terms, there are lots of different ones which have been created into a full ontology. Sequence ontology, SO, includes a lot of terms which are specific to variants. And if we just look at the brown ones here, which are related to transcription factor binding sites, which are regulatory sites in the DNA. So we might have the site removed, we might have the site amplified or duplicated. The binding site might be mutated. We might have a sequence variant within the site. And then we have, without reference to a transcription factor binding site, we might have more generic regulatory region variants, regulatory region removed or ablated, or the whole region amplified. So a lot of things there that you might include as annotations to your sequence variants. Here we have the, oops, here we have the stop gain, which I just explained. A normal coding codon is changed into a stop codon, leading to a shorter transcript. It seems my assistant is in the need of some attention, so he'll be taking part in the last parts of the lecture here, at least for a while. Now, the idea of changing an amino acid, this is something we already talked about when we discussed sequence alignments. We get a clue of the importance of an amino acid position in a protein by seeing how well it's conserved between different species. So conserved sites are more likely to be important and more likely to be pathogenic if they are mutated in a patient, whereas variant, highly variable sites are less likely to be pathogenic. So the algorithms that we have for, algorithms we have for the variant effect prediction might use the conservation data, but they also might look at the change in isolation without thinking about the conservation. But just a simple fact that if you are changing the tiny amino acid like glycine into a huge one like tryptophan, that is more likely to cause trouble in the protein structure because you can't fit the huge side chain in the same place. Or if we have a tight turn where the glycine is needed for being able to move the direction, change the direction of the peptide chain, the larger amino acid might not be there to allow that kind of a tight turn. If we have changes between hydrophilic and hydrophobic, or polar and non-polar side chains, that might interfere with protein folding as well, etc. And now these two algorithms have scales from 0 to 1, but they are kind of reversed, so that in the SIFT scale, number 1 means totally harmless, or a variant which is predicted to be harmless. Whereas in case of polyphen, the large values are probably damaging and the small values are probably benign or good in their character. So there's different terminology, but the coloring here between red and green is useful in figuring the quality as okay and not okay. And my apologies for the person who made these changes. If you are red-green colorblind, which is about 10% of male people, so you don't see the difference here, and that's annoying. We could have chosen a scale like yellow versus blue, which would be quite okay even if you have a color vision impairment. But because of traffic lights and other contexts where we have the red and green in our Western culture, so these have been so often chosen to be the go signal and the stop signal. Okay, we have variation data from a lot of species, but not nearly all. So it's just some chosen heavily investigated species there. And then we have some more in the ensemble genomes resources. There's a lot of variant data of the Baker's yeast, for example. And there are really many projects that provide data of variants. DBSNP is the largest database of single nucleotide polymorphisms. And then we have have other projects. We have the 1000 Genome Project, and even variants coming from PubMed are included if they are known to be pathogenic ones. And here's a link to the actual sources if you want to follow deeper into what these projects are. Okay, now in addition to knowing what the exact variant is and where in the genome it is, we'd like to know if it's a common variant or a rare variant. So we have a lot of data of population frequencies. HapMap project was based on haplotypes, and it was done before we had cheap genome-wide sequencing available, and now with next generation sequencing, high throughput sequencing, we have the 1000 genomes project, which, as you might have inferred, contains data of about 3,000 individuals. They realize that 1,000 is a small number when it's getting so cheap. So we have a lot of data coming from all over the world. And in addition to that, nowadays we have kind of single country genome projects so that people are trying to map the variants common in Finland or in Korea or Iceland or wherever. In the UK, the UK 10K has focused on sick people so that they have sequenced disease exomes. It means that they have done only exome sequencing, not full genome sequencing, and they have included the annotation that what was the disease in that individual so that we could try to find disease-associated variants from the data. Okay, the HapMap project had a lot of people, and as you can see that some of the Central South Asian populations are actually coming from the USA. There's a Gujarati population coming from, I'm not sure if this is Texas or Louisiana, anyway, somewhere in the US. And there's an Asian population also collected from the USA, and there's a European population, again from the USA, and so on. But anyway, the value of this project is that they really tried to go from the West European centric approach into a more global approach so that we would know what is the scope of variation in the whole world, not just in people of European origin. Okay, the 1000 Genomes project, well, here we see even Finland, and I think the dot should be actually on Tampere here in southwestern Finland, because that's the hub of the Finnish genome data. At least it was at the time of the 1000 Genomes project. Okay, it says 2,500 individuals, but now they've gone beyond 3,000 now. And sequencing at 4 times coverage is not a lot. It means that there will be gaps in each of these individual genomes, but it doesn't matter because you can align them. We have the reference human genome so that the sites of the variants can be identified. And this gives us a lot of data, as we'll see in a minute, that we can see which populations have which frequencies for specific variants. Okay, the reference alleles is a bit complicated. So the reference genome, as said, is not the genome of any individual person, and it is not the most common variants that are available. So for instance, in this position, we have a T, and even if it's a minority allele with only 5% of the people having it, because it was in the original sequencing data, it has been frozen to represent the reference sequence for that particular position. So having a variant which is different from the reference genome does not really tell you that you have a dangerous variant. You might in fact be carrying just something that 95% of the population has, which obviously can't be hugely harmful. And then if you look further in evolution, actually G is the allele in all primates, so it is the historically original allele, and this T is just a human-specific variant that is in a small amount of people. And even if we know that this T in this position causes disease susceptibility, so that it's disease associated increases the risk for something, so still it is the reference allele because it's what we had in the data. So T will be the allele that is present in the config and that is present in the reference genome. So T is the reference allele for this particular position. And G, even if the majority allele is called the alternate allele, just being different from that. And so the possible alleles or the genomic variants for this site in the genome would be T and G. And if we want to show this with a slash for the alternatives, they would be here, and in the reverse complement strand, we would have A or C as the corresponding nucleotides, and C would be the reference allele on the reverse strand. But now we get into complicated things. Now, when we do the translation, if this happens to be the coding strand, so the exon sequence has C as the reference allele and A as the alternate one. And so if this is the plus strand in the genome, in the chromosome, so in the database, we have T and G as the alleles. And in the gene, we have the alleles A and C, because the gene sequence is as the coding sequence shows it. And now, in dbSNP, they specify the strand so that they will tell you that you have A and C on the negative strand and T or G on the positive strand. In PubMed, depending on where the data comes from, so you might see A to C variant or T to G variant without necessarily specifying which strand they are referring to and whether they are looking at the gene or not. Obviously, in the cases where we have a coding SNP, which is in the positive strand, so then the alleles in the database and alleles in the gene would be the same, and there is less chance for confusion. But just to give you the warning that you really need to check the source of your data if you want to investigate a specific variant. You need to know which strand it refers to. Okay, so let's do a quick demo. Let's find a specific variant in one gene. I'm just copying the number here. We don't have time to look at the variant effect predictor yet, but that will be on Friday. Come on. I'm going to shortcut and I'll be looking for just the variant code. while it's searching, I might as well go for MCM6, the human gene that we were supposed to be looking at. Is this correct? MCM6, yes. Okay, so here we have data on this SNP, and if we look at the gene region, we have a very short summary of the variant data here at the bottom of the highest zoom factor square. So we have all variants here, which is pretty much almost every site has a possible variant. And then we have phenotype associated variants here, which are not that many. But looking at the variant information here, so we can see that it is located in the region we were interested in, chromosome 2, 135 million. Yes, it's here. And this one has multiple alleles. 3 ancestral one is G, and then we have a high percentage of A, and C is a minority allele. We have links to clinical significance. There's an association with, I believe this is related to lactose intolerance, or rather lactose persistence. And if we look at the description here, so even if this is not in the lactose degrading enzyme, it's in a neighboring gene, this MCM6 variant has also an effect on this. Okay, so we have phenotype data showing that we have association to body mass index, hip circumference, and then here we have lactase persistence. This is a link to the clinical variant data, and it's coming from the online Mendelian inheritance in man. You may have heard of most people in the world not tolerating lactose when they are adults. So lactose is okay when it's coming during the period of lactation, but when a child grows up, the lactose becomes something that is not digestible, which will lead to diarrhea and gas problems and whatnot. But in Western Europe and in African regions where keeping cattle has been really common, so it's a huge advantage if you can use the cattle milk as a source of nutrition and have the lactase enzyme persisting. So lactase is the enzyme breaking down lactose, and usually it's not expressed after the age of 12 or 16 years, and you become lactose intolerant. But when the lactase is persisting to the adult age, you can use the milk as a source of protein and as good quality nutrition, and that gives you an advantage. And that's why this lactase persistence phenotype has become really prevalent in many European populations. In Denmark, I think the number is like 97% of the people being able to digest lactose, whereas in Thailand, it's the opposite. Just a few % of the people can consume lactose in adult age. So it's a fairly interesting variant which we have here. And let's see if we have the population data here. I just go back and population genetics. This should give us graphs for many different populations. So here we have the bar graphs, and we should have the pie charts coming up. So you can see that what is the frequency of each allele in populations. So in European populations, we have a lot of the alternate allele, which is the one leading to lactase persistence, whereas in East Asian populations, it's basically zero, and it's mixed in many populations. Okay, it's taking time to load the data. We would have more graphs there, but they are not showing in a reasonable time. So we might as well stop here and leave the variant effect predictor as the last topic of variants for Friday. And on Friday, we also have a chance to look at the answers to your homework 3, which is related to variants, to get a little bit more of a revision of these topics. Okay, I guess you were referring to my dog, not myself, and indeed he is such a cute dog. His name is Gaudi, and he is 8 months old, so pretty well educated at the point, but still a cute puppy. Okay, any questions related to the actual topic of variants we've discussed today? Okay, not really. Thanks for being here.